so when we thought about this wound signal, we have further categorized this into electrical, physical, and the chemical signals, and these signals are propagating to the distal part of the plant. So we try to understand this signal propagation with a very simple model. Here you can see that as soon as there is a herbivore wounding happens to the local leaf, so there is a this is the xylem vessels which conduct the water in plants, and these are phloems which conduct this food for the plant. So as soon as there is a break of this leaf tissue, so this generates a pressure changes along the xylem vessel, and this pressure is also transmitted radially to the nearby cell of the xylem and transmitted to the phloem cells. So this pressure changes, this leads to the activation of a protein called glutamate-like receptors, and this activate this protein, and these are kind of ion channels which leads to a huge ion fluxes. And possibly here we can see that calcium is playing a big role in this ion fluxes, and this ion fluxes leads to a depolarization of the cell membrane. And here you can see that when the cell membrane is depolarized, they take some time to come to the vessel level. So during that period also these endomembranes are depolarized, and this might lead to activation of several genes which is there in the endomembranes in some organelles. So what we know that maybe possibly this activate the lipoxygenase enzymes and OPDA release, which is related to the a synthesis of another hormone which is called jasmonic acid. This is a very important hormone which play a crucial role in the plant defenses. So so here we see that there is a membrane potential changes, there is a pressure changes, and there are some hormones traveling together in this xylem and phloem cells, and this leads to the activation of the defense responses. So looking more closely how these signals look like, during my postdoc work, we were analyzing these signals more closely. So here I will show you a very nice video in which you can see. So this is a Arabidopsis plant, and this insect Spodoptera is feeding on this, and we have some electrodes put on the distal leaf which is connected to this leaf, and you see this is a vessel level, and as soon as insect break this midvein, we see a huge depolarization of the membrane, and this depolarization remains for the several minutes, and then they come to the vessel level. So to correlate this with the some ions, we also visualize this calcium ions which we predicted. So here we have transgenic Arabidopsis plant which has GCAM for this cytosolic calcium measurement. So you see when in distal leaf, when there is a membrane depolarization, at the same time we see a upregulation of cytosolic calcium. Here we have measured that cytosolic calcium, and this depolarization of membrane and activation of the increase of cytosolic calcium coincide with each other. So we at this moment we cannot say that which one is first and which is regulating this defense pathway, but we know that both exist. This calcium ion fluxes is there, and there is a huge depolarization. So there must be several other ion channels or pumps are there on the membrane which is leading to this huge ion fluxes. So leading to this like observation, right now we have several questions with us, like how these signals are generated, propagated, or decoded, and whether these signals are correlated. So right now we cannot say that the calcium or electrical signal is completely correlated, and how if they are there after wounding, how they leads to the plant, defense responses. And can we say there is some tissue, a specific tissue, which is responsible for this responses? So so far, whatever people have tried to understand in this field, so here is the summary, like what genetic component we have understood so far. So I will briefly explain here. So this is a wounding. So when herbivore is there, this wound has happened, and this leads to the increase in of the glutamate in the apoplast. At the same time, there is a pressure change which we saw earlier, and this activate this activate the stretch the membrane, and this activate the mechanosensitive ion channel, which is a MSL10. And very recently, people have found its role in the defense responses. So this membrane stretch activate this MSL10, and this leads to the membrane depolarization, and the membrane depolarization and glutamate accumulation in the apoplast. This leads to the activation of the GLR proteins. And this GLR is again like this leads to the huge cytosolic calcium fluxes. And during my postdoc work, I found that another genetic component, which I told you, there is a plasma membrane proton pumps. So there are several members of this proton pumps, and I found that this plasma membrane proton pump 1, which we call AHA1, this is also like cytosolic calcium inhibited this proton pumps, which increases the repolarization phases. So if they inhibit the proton pump calcium, then what happens that the membrane remains in the depolarized phase for very prolonged time, and this leads to the again to the activation of GLR and this increase in cytosolic calcium. And further this through two different pathway, they upregulate the reactive oxygen species and the jasmonate pathway, and leads to the defense responses. So we also know few other components like CNGC19 and NXine recently, who also play a role in the defense responses. So now, based on all this research background, when I moved to India, I tried to define my lab objectives around two major area. So first one is exploring the identifying the new genetic player for defense responses, and for that region, I'm using natural variants of Arabidopsis. And then I also started working with the Chickpea, and we are analyzing these differential defense responses in the wild and cultivated chickpea, and we are using several omics approaches to understand more about this. And the third one is identifying the specific cell population, which is more responsible for this herbivory responses or in wound responses. And for that region, I'm using AHA1 identified proton pump as a tool. And the fourth interest after coming to India was to understand more about this insect host preferences. So I think like evolutionary, when plant and insects were evolving together, they have insects had decided to feed more on one type of plant compared to another. So using that approach, we are trying to understand more about this insect aspect of plant interaction. So now I will briefly tell you few results in this area when I moved here. So when we are looking for this specific cell population, what I did, like I found earlier that AHA1 genes, they are expressed in the both tissue type of this plant, like xylem and phloem cells. And this is also known that this protein, this AHA1 protein, this has a C termini, and there are several phosphorylation sites on this C-termini, and people think that several of these environmental signals might be integrating and this C-termini. So my approach was to truncate this C-termini and make a constitutively active proton pump. And here is a example, like I will show just a phenotype briefly. So here you see this, when you have this AHA1 gene absent in the wild type plant, in this Arabidopsis plant, you see that plants are smaller. And when we complement this gene with its own promoter, which expresses in the xylem and phloem, then they look similar to the wild type plant, Columbia 0 plants. So we try to target this in a specific cell, which is a phloem, using the GLR 3.3 promoter. And here phenotypically it looks they have complemented but partially complemented this plant growth. And then we made a constitutively active proton pump by truncating this C-termini, which we call AHA1 delta 844. And when you just target this in a phloem tissue, the plants looks bigger. So this shows that this proton pump might be constitutively active there. And further, we have expressed this active proton pump in several other tissue type like xylem and all, and we are analyzing them for the herbivory responses. And next, coming to the Chickpea project. So here one of my students is working on this Chickpea, like differential defense responses in Chickpea. So we have collected several accession of Chickpea and tried to understand if we have different feeding behavior on this Chickpea accession. So here you see, compared to the artificial diet, these insects are very healthy. And when they feed on the chickpea plants, so this is a susceptible variety, and so in susceptible variety, they have a bit better growth compared to the tolerant one. And so based on this data, we further analyzing their defense responses using different markers and utilizing the RNA-seq approach to understand the different genes. And I'm recently I was also able to establish an electrophysiology lab in our group, and we are able to measure this surface potential on the chickpea, and we are exploring this aspect, how these early signals, calcium and electrical signals and ROS are there in the chickpea. And coming to the fourth objective, where I told that we try to explore the idea that the insects have preferences for the food. So when we thought what happens, like why insect is preferring one plant compared to the others. So we use Helicoverpa armigera as insects, and we fed them on artificial diet and on the host plant, which is a tomato plant here, and then on the non-host plant, which is a capsicum for the Helicoverpa armigera. So we found that on the non-host plant, their growth is very less compared to the tomato and artificial diet. So so then we analyze the oral secretion of these insects to understand because there is always a continuous chemical dialogue between plant and insects. So insects, when they are feeding, they are releasing oral secretion to the plant, and the plant, as a defense, they are synthesizing secondary metabolites. So this might be going to the insect as well and hampering their growth. So after feeding on these different diets, we analyzed the oral secretion, metabolites of this oral secretion of Helicoverpa armigera. And here you see we have some common components, but at the same time, there are several metabolites are accumulating differentially in these different diets. And in case of artificial diet, there is more phospholipids. In case of host plant, tomato leaf, there is a more alkaline alkaloids, and in case of non-host plant, there is more terpenoids. So then we analyzed like their differential, their fold changes, like how much they are accumulating. And recently we have published this work, and we found that few this metabolites, like here you see chlorogenic acid is accumulating more in case of host non-host plant. And when we fed this chlorogenic acid to the insects through the artificial diet, we found that there is a dose dependent growth inhibition in case of chlorogenic acid. So right now we are exploring more in this area also, and we'll try to understand through proteomics and other approaches, and know the specific components in the oral secretion which might act as a defense activator in plant or suppressor in the plant, and this could be utilized as a sustainable crop protection strategy. So at the end, I would like to thank CSIR NCL for supporting me as a Ramalinga Fellow and start my lab there, and all the members there, and definitely Professor Edward for supporting my postdoc research at University of Lausanne, Gettengloger, and the funding agency DBT, and yeah, I'm looking for the potential collaborator on this topic and explore more in this area. So I think, Ratna, maybe I finished the time. Should I go to the next topic, or?
Yeah, if you can briefly discuss your journey slide.
Yeah, so initially when I saw these questions, 4 questions, so I thought of picking this women aspect, but then I realized no, because I'm here in India for 1.5 years, and it was really a great learning period for me to really adjust to this system and everything. So for me, the first thing was because I was for a very long period I was outside, and the punctuality distress was there with me. So like I found people like and not a specific area, but everywhere, like they don't stick to the time. So that was a kind of so you have to deal with this, like you need to slowly adapt with this. And so we have as a new faculty, you have very limited funds. So my advice to the new faculties is keep looking for the more funding opportunity that really helps you to explore more things. And the third thing is establishing new facilities. So if you are joining some lab and you are looking for something totally new, like for me, electrophysiology was like introducing a new area in the NCL, and it was so difficult. Like it took 1 hour to figure out vendors and suppliers, and you need to be really patient with this. So my advice to the people is definitely you should look for your passion. You try to get new facilities according to your requirements, but at the same time you try to develop few objectives, research objectives, which align with the available resources. So you don't stop completely your research work. So you keep doing some research, and at the same time you can develop the facilities. And the last thing is hiring good candidates. So when you are establishing lab, you have a lot of responsibilities, like enormous things will come. This for me, the several things was new, like handling the project money, then dealing with the office people, then vendors and all those things. So the best advice would be find some person initially, at least one person who has some experience, practical experience of research. They will really help you to keep running your research objective while you are busy settling your new lab. So yeah, and keep exploring, like don't hesitate to contact other researchers. Be always like try to adjust with the system. Like it's not like in one day everything will change. You need to change a little bit, and then accordingly maybe people will fall according to your nature. So yeah, this is all. Ratna.
Well, thank you, Dr. Archana, and great listening to you. And also, I really like the last point: listen to others, but follow your judgment. That is some great advice.
I think that's a normal advice. So yeah, you definitely like talk to several people. I did the same. I talked to several people, but don't get opinion immediately. You also experience few things with them and then make your opinions, combining both.
Yeah. Okay. Yeah. Okay. Maybe questions for Dr. Archana? Anyone from the group? Okay, I have one question for you. Is it a very, sometimes you see a species-specific response between a plant and a herbivore? That's the first part. And the second part is the specificity depend on what kind of herbivore it is? Is it like in a one which is a host and herbivore reaction, or is it the one which is more dependent on a mutualistic reaction?
So yeah, I would say like it's really dependent on the type of plant you selected and the insect you are working with. So as I mentioned you, here I used Helicoverpa armigera. It's a polyphagous insect. So they eat completely on anything in a given condition, but still they have preferences. Like they have some priorities. If there is a availability of tomato and capsicum, they will go to tomato, but they can eat on capsicum as well. So yeah, definitely this interaction, plant and insect interaction, completely depend on plant species and on the herbivore, like type of herbivore. So basically they have evolved for millions of years and they have adapted to some kind of so insects have also evolved, plants have also evolved. We have several new insect species also developed during this evolution process. So yeah, it's totally depend on what system you choose.
But the
you have to be very cautious.
Right, but the same plant can have different mechanisms for reacting to a herbivore, right? Or no?
Yeah, mechanism, I would say like yes. So in case of, suppose I'm just giving example, there is not a proof. So it might be possible that when your Arabidopsis your chickpea plant is fed by Helicoverpa armigera, and when it is fed by Scodopedaria, they will have a different response. And so from where this response are coming, basically it's going back to their initial activation signaling, like how these signals have generated. There might be some changes. There is a upregulation of calcium, maybe depolarization of membrane, but with different intensity. They have less upregulation of calcium, and that's why there is a differential responses.
Okay. Okay. Well, thank you. Are there any more questions from the group? Okay, if not, then Dr. Archana, really thank you. I will request you to kindly unshare the screen.
Yeah, sure. Thank you, Ratna.
Thank you. Okay, so I will call now Dr. Chitralekhang Das Gupta. Dr. Chitralekhang, can you hear me? Oh, you are on mute.
Yeah, now am I audible?
Yes, you are audible. Can you just share your screen then, just making sure everything is okay?
Yeah, sure. Is it okay?
Yeah, looks good. Okay, so I welcome our next speaker for the life sciences other session, Dr. Chitralekhang Das Gupta. She is an assistant professor at the Amity Institute of Biotechnology at Uttar Pradesh. Dr. Chitralekhang, your time starts.
Yeah, well, Thank you so much, Dr. Ratna. And I'm really privileged and honored to got this opportunity to connect and present in this platform. I'm really thankful to the organizer. Today, I'm going to talk about the molecular insight of arsenic remediation by freshwater microalgae Synetesmus quadriquata and production of bioenergy carriers. So I will go in a very brief manner. I will not going to show all the detail of my research data. But first of all, shall I make it full screen?
Yeah, if you can make it full screen.
Yeah, sure.
Yeah, works. Thank you.
Okay, so so first, okay. Yeah, now it's okay.
Yeah.
So first question is why algae? Why algae is in the prime focus nowadays? Why we are using algae for different purposes like bioremediation, biofuel production, and many more, right? So what are the benefits to using this algae? They are mostly non-pathogenic. They are mostly non-pathogenic, easy to handle, eco-friendly to the environment. There's less limitation due to the seasonal changes. Ability of strains and biomass throughout the year can be grown in a small space like photobioreactor or open pond with high biomass productivity. The utilization of algae for arsenic bioremediation has an edge over different conventional technologies that we are using like flocculation, oxidation, and membrane filtration. Nowadays, we are using RO filtration. So those different type of conventional technologies and also different biological agents, those are capable of remediate arsenic like microbes, fungi. So, but it has some benefits. Though the microbes and fungi are involved in the biotransformation of organic species, but their efficiency very much dependent on the physicochemical parameter of water and soil. They might be pathogenic and byproducts may not have commercial values. But for algae, we will get the byproducts, the biomass we will produce, maybe can be utilized for other purposes, and also they are mostly non-pathogenic. And they have large surface to volume ratio so that it can rapidly absorb the nutrients, metal uptake capacity in both high and low contamination, accumulate and metabolize all the types of arsenic species present in the water or in a soil. They are ubiquitous in nature, and that means you can find everywhere. And they have versatile metabolic pathways and production of different value-added biomolecules and energy carriers. Biomass can be utilized for value-added products like biofuel, biofertilizer, pharmaceutical and nutraceutical products. And nowadays, the genome sequences are available for many of the algal species, which actually help us to do some genetic engineering work or genetic manipulation, targeted genetic or metabolic manipulation. So there are so many benefits, that's why we are using algae nowadays for different purposes, not only bioremediation, and mostly the focus now is on biofuel. So there are three important domains of research in algae. First is the bioremediation part, where we can also talk about the arsenic detoxification, arsenic removal or detoxification from wastewater. But when we are using wastewater, it has dual benefits. Algae also get the nutrients from wastewater and it will grow. That means we will get the biomass of algae, as well as during growth, it will remove the contaminants from the wastewater. So this is very important aspect. Another is the molecular aspect, that is now in algae, as the availability of different algal genome sequences, we have started different molecular like metabolic engineering, genetic engineering, and also we are transferring different genes to the other organism. So this is another important aspect. And third is the bioenergy. That means we are producing different energy carriers from algae. We are getting biodiesel, we are getting biohydrogen, bioethanol. So this is another very important domain of algal research. So we have done genome sequencing of a particular microalgae, freshwater microalgae. The name is Synodismus quadriquada. Why we have chosen this algae? Because we have screened so many algae for lipid production and biomass production. And we have found this algae is very important and very potential for biomass productivity and lipid yield. So we have chosen this algae and we have done the genome sequencing for this algae. And actually which opened, it's a kind of treasure box which open up so many important gene sequences for us for just to work on. We have now so many avenues opened up with us to work on. So that is very important. We have found out almost 13,514 genes in de novo sequencing, and even more, that is 16,739 genes in reference guided annotation. References for reference guided annotation, we have taken another freshwater microalgae, that is Monoraphidium neglectum. So with that, we are getting so many genes identified. And for lipid production, we have identified 283 genes in their pathways. After that, we reconstructed the metabolic pathways, and that after reconstructing this metabolic pathways, we have identified the genes for arsenic detoxification. We have identified the genes for TAG biosynthesis, that is triacyl glycerol, that is lipid production. So triacyl glycerol biosynthesis pathways, genes are also identified. We have identified the Fe hydrogenase, that gene is responsible hydrogen production. Hydrogen is another energy carrier. So also we have identified some genes that is having the cellulitic activity, that is cellulases. We have identified, and it's very rare. It's the second report of the cellulases, because first report in Chlamydomonas, another microalgae, and then we have reported, not yet reported, we are actually communicated this paper. So this is another second report of the presence of this gene, and also the nitrogen fixation gene, and different many more. So it's like we have now different avenues to work on. So we started to work on in all the avenues. So in arsenic contamination, what is arsenic? Arsenic is a chemical element or metalloid which is very toxic. And in arsenite, it can in nature we'll find the inorganic trivalent arsenite and pentavalent arsenate. These two species are very predominant in water and soil, and they are very toxic. And they are toxic means they can cause cancer, skin lesions, different cardiovascular diseases, diabetes, death. And the limit of arsenic in drinking water is only 10 microgram per liter for according to the WHO guidelines. Okay, World Health Organization guidelines, we can only permit this limit. But millions of people around the world are exposed to the arsenic at a concentration of much higher than the guideline value. Okay, so this is a recent report in Geoscience Frontiers. They have shown how the world is affected with arsenic. They have found that 180 countries, they have reported 108 countries are affected by arsenic contamination in groundwater, and highest among them are Asia, Europe, and followed by the regions like Africa, North America, South America, and Australia. And more than 230 million people in worldwide, which include 180 million from Asia, are at risk of arsenic poisoning. So this is very important topic which we need to really focus on. So what we have done, we just collected the sewage from the sewage treatment plant present in here in our Lucknow only. So this is actually Asia's biggest STP. We situated in Varwa village near Lucknow. So where from there we collected the water and we tested with different algae for the remediation of arsenic as well as other metals also. So not only metals, we also tested for other contaminants. But by focusing arsenic, I can show you that is we are finding that the algae can remove or algae can absorb arsenic very well, and we are getting almost 74.8% of removal of arsenic from that sewage water. And going to the genome sequence of Cinerismus, we are also find some important genes which can be which you can say that is responsible for this absorption of arsenic. We have found that heavy metal transporting ATPase, arsenic transporting ATPase, arsenite transporter, another family of protein, that is, and also multi-drug and toxic compound extrusion genes, heavy metals, exflux system, and most importantly, the gene that is arsenic methyl transferase. This gene is very important for the methylation of arsenic species to make it less toxic, because after methylation, the arsenic species will be less toxic to the inorganic forms. When we blasted that sequence in NCBI, we have found it has similarity with different other methyl transferases of bacteria as well as few algae, which already reported in NCBI. So we align them and find some conserved region in their sequences. We aligned with other 4 algae, and we have found this is our algae, the Cinerismus quadriquada, and we aligned with other 4 algae reported in NCBI, and we found the conserved region for the same. And also we checked with a software, that is Sci-ROI software, whether it has any similar crystal structure present in the web portal. So we have found that it has similarity, almost 77% of coverage and 100% confidence with the X-ray crystal structure of another methyl transferase, arsenic methyl transferase from Chlamydomonas renarditi. It is another algae, and it is a model algae where so many molecular work already have been done. So this algae, we have found this similarity with this algae. So now we have to work more, but before that, we just speculated the pathway, the gene we have identified, and the data already reported in different publications. We just gather all the data and our data. So we've speculated a pathway of arsenic detoxification in microalgae. So what we speculated that algal surface binds and absorbs the arsenic species. There are so many functional groups are present, carboxyl, hydroxyl. So these groups are actually helps to absorb this arsenic species. Those are mostly inorganic arsenic species with trivalent and pentavalent inorganic species that has been observed inside the cell of algae. And then there is a oxidation and reduction. Maybe there may be presence of oxidase and reductase, which can oxidize and reduce this arsenic species. And after that, there is a methylation, maybe a methylation, possibility of methylation, which can produce different methyl arsenicols like monomethyl arsenic acid and dimethyl arsenic acid. So these are all assumption, and in some algae already they have been found. This type of methyl arsenicols are found, and there is another trimethyl arsine. This actually volatile. So I'm going one by one. So this is the oxidation reduction reaction. Sometimes they can be formed complex with thiol group and sequestered in the vacuole and effluxed. And there might be some methylation and formation of different methyl arsenicols, which are less toxic than the inorganic forms. And lastly, they can form also volatile methyl arsenicols, volatile arsenic species, which can be volatilized from the cell to the environment. So by this type of method, it can absorb the arsenic and it can remove the arsenic from the water. So sometimes we don't know the mechanism how, but we have also found in some literature that algae can produce arsenolipids, different complex arsenic compounds, which can be taken by some aquatic or marine animal or fishes, which can also produce arsenobutane. After production of this arsenobutane, the microbial metabolism will again convert it into inorganic, trivalent, and pentavalent arsenic species. So by this cycle can be completed, and also we will get some arsenic absorbed from here, and it can go through the methylation part, and this all methylated species, arsenic species, are less toxic, and some of them can be volatile and can goes out to the environment. So this methylation, why it is very important, because we have already found a gene that is arsenic methyl transferase. So methylation actually reduces the toxicity, that is already I have told you. And higher plants may not have this ability to methylate arsenic. So we can transfer, this methylated arsenicols in the plants, whatever the reports we are getting, actually may be originated from the microorganism present in their rhizosphere. So we can transfer this algal gene and express in the higher plants, like in rice, maybe, which is very common food for us, common grains for us. So we can transfer them, this methyl transferase gene, to the other plants. So that is a possibility. So that's why we are focusing on this particular gene sequences. Now, lastly, when we are using different wastewater like sewage or any other wastewater, ultimately we are producing the biomass. So what to do with this biomass? So after producing this biomass, we can also produce different other energy carriers from them. Like we can grow the algae in a wastewater, and then we can harvest them. We have this raceway pond we have in Lucknow in NBRI, CSR NBRI. So we have grown the algae in raceway pond with the sewage, and then we harvested the biomass. Then we extracted the lipid, and then we esterified to produce biodiesel. After transesterification, we are getting the biodiesel, and that biodiesel, after gas chromatographic analysis, we have found that they are mostly predominant with C16 and C18 fatty acids. So again, with mass spectrometry, we are find many other different molecules which maybe have some benefits for development of different drug molecules, like this eicosapentonic acid. This is actually omega-3 fatty acids. Some phytol also we have found. So different biomolecules we are getting from that biomass. So in gene sequences, when we reconstructed the metabolic pathways, we have also found there are two places where we can target for overexpression of that gene, because we are getting very less number of gene homologues, and maybe that is the reason why we are getting less lipid yield. Because all of you know, though we are working on algal biofuel for a long time, but still the researchers are not getting the perfect result for the commercialization because of the economical problem or cost related problem. So we have to somehow increase the yield of the lipid. So for that, we can do some metabolic engineering. So we have identified 2 targets where we can overexpress that enzymes to ameliorate or increase the lipid concentration. This work actually we are going we're doing now with a project of SRB. And also we can get biohydrogen from algae also. That biomass, after production of this biomass, if you develop an anaerobic condition for the algal growth, we can also produce biohydrogen, because hydrogen can be produced in anaerobic condition only. So anaerobic condition we have to maintain somehow. For that, we deprive the sulfur in the media, so that anaerobic condition will start, it developed, and hydrogen will produce. So we develop different photobioreactors to produce hydrogen, and we are getting hydrogen also from Synedesmus, and as well as one another algae is chlorella. So we are getting about 17% of volume by volume hydrogen continuously for a 7 days experiment. So this hydrogen, after getting this hydrogen, the residual biomass, that is also useful, because when we are depriving sulfur in the media, the cells are in stressed, and in the stressed condition they will produce more lipid. This is the normal nature of cell. If you keep them in a stress condition, it will produce more lipid. So ultimately the residual biomass, that is also rich in lipid. So from residual biomass, we can also produce biodiesel. So it can be, the biomass can have dual function. It can produce hydrogen, as well as we can get the biodiesel from the residual. So by this type of experiment or this type of method, if we can improve, we can really lower the cost of the production of any energy carriers. So coming to the brief achievements of my research work, we have identified 2 different algae which are very efficient for the treatment of STP, sewage from STP water, STP, and it improves the DO always, because if you grow the algae in any water, it will improve the DO by releasing oxygen inside the water, and subsequently reduce the TDS, BOD, and COD and toxic metal, including arsenic. Microalga Synedesmus quadriquada was selected as a preferred organism because we have screened many algae for that, but we selected this because of it has high biomass and lipid content. And Synedesmus has shown maximum evolution of more than 17% of hydrogen. And after releasing this hydrogen, the residual biomass, it has high lipid content. So we can also use the residual biomass for lipid production. Also, we have the first report of genome sequencing of Synedesmus quadriquada, predicted the genome size of 65.3 MB, 35 MB with 13,514 genes. Already I have told you, and even more in a reference guided annotation. So we have identified arsenic detoxifying genes. We have identified the bottlenecks in TAG biosynthesis pathway, that is lipid biosynthesis pathway, and we started the targeted genetic manipulation for this pathway. And also we have identified the Fe hydrogenase gene responsible for hydrogen production. That is also the first report for this alga. And cellulase belongs to the GH1, GH5, and GH9, 10 families that are present in higher sequence identified with the enzyme from invertebrate, fungi, and bacteria rather than cellulase from. That is also important, because the most of the enzymes we are getting, that is more similar to the lower like bacteria, fungi, invertebrates rather than the higher plants, because the microalgae are more similar to the prokaryotic organisms and lower group of organisms than the higher plants. So these are some selected publication from of my publication book chapters, and this is a book is on biohydrogen production. My journey as a woman scientist, I started my PhD in Calcutta University, and I completed in 2005 and awarded in 2006. After that, I moved to Paris, France, for a postdoctoral research. But after that, I got the opportunity to do postdoctoral research in another institute in Berlin, in Germany, that is Max Planck Institute for Infection Biology. But I couldn't travel due to the birth of and infant care of my first child. So I had to take a career break that time. And then with that child and all my family issues, I have to return. And then I continued my research work in IIT Kharagpur. As a research associate, I joined the Professor Debabrata Das lab in biotechnology department. But then again, I have to take another break due to birth of my second child. Then with so many breaks, I was like, what to do next? How can I start my research career again? So that time I got the opportunity, thanks to government, Indian government, that they have some policies for the women also. So I got the opportunity to carry out my research work again as a DST woman scientist, and I joined CSIR-NBRI Lucknow in 2012. Then after completing the 3 years tenure, then I was again stuck. Then fortunately I got CSIR pool scientist and continued in CSIR-NBRI. So I continued my research in NBRI almost 7 years. After completing CSR pool scientist, again I was like, I was stuck to do what to do, how to proceed for the next research career. So then there's a interview where I saw shortlisted in NBRI, but unfortunately I was in waiting 1 for the principal scientist position. Then I was really stuck at that time and don't know what to do. So that time didn't have any idea how to carry out my research for further. So that time really I'm thankful that I got the opportunity in Amity University to join as assistant professor. Then I got the platform to start my research again in 2020. But that time it was really hard because that time it was locked down for COVID-19. But still I'm trying, and fortunately this year, the last year in 2021, I got a project from SCRB on this topic for biofuel production to metabolic engineering for the pathway of TAG biosynthesis pathway of microalgae. So I have started my small lab, and 1 PhD student has joined. So I'm still struggling, but I'm doing, I think I can overcome the situation, and I can do much better maybe in the future. So I'm still submitting different projects to get more projects to establish my lab and continue as a research scientist also. So thank you all of you, and thank you Ratna.
It was very nice hearing your talk, Dr. Chitralekha, and many thanks for also sharing your journey as well. We greatly appreciate that. Any questions from the group, Dr. Chitralekha? Okay, while we are waiting for questions from others, I have a question. In one particular part, you mentioned that most of the, arsenic methylases should be, that the trees are getting are maybe from the microorganisms, and you mentioned the rice, but that may be only in the case of agricultural crops. Do you think that's also the case for like naturally grown trees, because there's so much arsenic in soil? So
yeah, actually till now there is no report for arsenic methylase for higher plants.
Okay.
So maybe, yeah, that's the case. That's why the researchers, what they are doing, they are identifying the arsenic methyl transferase from different microorganisms and maybe from fungi, and also nowadays from algae. So they are transferring this gene to the higher plants to methylate the arsenic. This is a really very interesting, because all of the organism having this methyl transferase even in human. But why the higher plants, we are not getting any link to the higher plants. We are not getting any reports for the higher plants. So that is really tricky. So that's why we are doing.
It's surprising, because you get plants in aquatic, terrestrial, and arsenic is kind of there both.
Yeah, sometimes the scientists, they reported methyl transferase from higher plants also, but they didn't find any sequence or gene sequence. So they cannot trace that particular sequence or gene in that particular plant. So sometimes they speculated maybe this methyl arsenic species is coming from the microorganism which is present in the rhizosphere. So that is a speculation. It's a lot of research need to be done in this particular field. Actually, lot of research need to be done in the field of algae, right? So we have lot of reports for bacterial system. We have lot of reports for plant system also. But in algal system, for the for molecular aspects, we have to work a lot. We really just, you can say it's a beginning. So we need to know. Yeah. Okay. So we are nowadays we are having so many algae which are the genome sequence is known, but still it is very less compared to the bacteria and higher plants. So that we have to actually do.
Okay. Well, thank you, Dr. Chitralekha, and also I thank both of you, both the speakers, Dr. Archana and Dr. Chitralekha, before wrapping up the session for this breakout room. I request all of you, the other breakout rooms, the session is ongoing. Please do join whichever fits in your preference. And thank you for being great presenters at the Sci-ROI event.
Oh, thank you. Thank you so much, Ratna.
Thank you, Ratna.
Thank you.
Thank you, Ratna, for the opportunity. It's really nice.
Yeah, sure. It's really, we are privileged and really honored to connect with this platform.
Well, we are very happy to hear that. It's our first event, so we are very happy to hear that.
Yeah. Thank you. Thank you so much.